Welcome everyone. I'm here to take about affinity chromatography technique. It was first discovered by Pedro Cutricasas and Meir Wilczek, and it is a biophysical technique that enables the separation, identification and purification of the components, both qualitative and quantitative analysis. And it is a technique which involves mobile phase which carries a mixture, and it is caused to move in contact with the selectively absorbent stationary phase. And this is a type of liquid chromatography. An example will be enzyme with an inhibitor, antigen with an antibody, etc. And this affinity chromatography is a method for selective purification of a molecule or a group of molecules from complex mixtures based on highly specific biological interaction between the two molecules. And this interaction is typically reversible, and purification is achieved through a biphasic interactions with one of the molecules, that is ligand, which is immobilized to a surface, while its partner, that is the target molecule, is in a mobile phase as a part of a mixture of complex. So the capture step is generally followed by washing and elution, resulting in the recovery of highly purified protein, and highly selective interactions allow for a fast, one single step process with potential for purifications in the order of several hundred to thousand folds. And the recent decades have been tremendous advancement in utility of affinity chromatography, with development and support materials such as flow-through beads, magnetic beads, and monolithic materials, as well as new ligands with a variety of interesting biological properties. In addition to this approach is no longer used only for purification of specific biomolecules. It is also quickly becoming a method of choice to study biological interactions, and can be used for the preparation of samples for mass spectrometry or for specific removal of contaminants. So what is the basic principle of affinity chromatography? So it contains a mobile phase and a stationary phase, and the mobile phase will be always liquid, and the stationary phase consists of a support medium where ligand bound to the targeted molecules. And as the crude mixture of the substances is passed through the chromatographic column, the substances with binding site for the immobilized substrate bind to the stationary phase, while others were eluted out from the column. And the bound target molecules can be eluted by displacement method. So what are the components required for affinity chromatography? So first one will be matrix, and second one will be spacer arm, third one will be ligand. So matrix, it is an inert support to which a ligand can be directly or indirectly coupled or bound. So it must be insoluble in solvents and must be chemically and mechanically stable to work with it. And it exhibits good flow properties, and most useful matrix materials are agarose and polyacrylamide gel. And the spacer arm. The spacer arm is connected between this matrix and the ligand. So it is used to improve binding between ligand and the target molecule. And ligand, it refers to the molecule that binds reversibly to a specific target molecule, and this ligand can be selectively only after the nature of the macromolecule to be isolated is well known. Where a hormone receptor protein is to be purified by affinity chromatography, the hormone itself is an ideal candidate for the ligand. And for antibody isolation, an antigen or haptor may be used as ligand. If an enzyme is to be purified, a substrate analog inhibitor may be used as the immobilized ligand. And this is the steps involved in affinity chromatography. First one will be the preparation of column. The column is loaded with a solid support such as saccharose, agarose, cellulose, etc. and the ligand here is selected according to the desired isolation. That is what we are going to use for the separation. And the spacer arm is attached between the ligand and the solid support. The next comes loading of sample. The solution containing a mixture of substances is poured into the elution column and allowed to run at a controlled rate. And elution of ligand molecule complex. The target substance is recovered by changing conditions to favor elutions of the bound molecules. While the first example of the affinity chromatography exploited binding of the enzyme amylase to insoluble starch, and it was the development of solid support materials and the chemistry used to attach ligand to these solid supports that truly enabled affinity chromatography. So since these seminal advances, there has been an explosion in the development of more support materials like affinity ligands and improvements in the method that make affinity purification an attractive approach to purify biomolecules from the complex mixture. And this is the structure of affinity chromatography, where it shows the column in the left side. That column contains the stationary phase and the mobile phase. In that only we are introducing this matrix, spacer arm and the ligand. These three are interconnected, and these three complexes will go and bind to the target molecules like enzymes or antigen, as I informed you in the previous slide. And once it binds, it gets eluted from the column by the displacement method. So it is used for purification of enzymes and separation techniques, etc. Then applications of affinity chromatography. So applications include separation of mixture of compounds, removal of impurities or in purification process, detection of substrates, investigation of binding sites of enzymes. Then additional uses of affinity chromatography include the ability to concentrate substances present at low concentration and the ability to separate proteins based on their biological function, where an active form can be separated from the inactive form or a form with different biological functions. Thank you.